Presentation share हो रहा है?
Yes, ma'am.
So going back to the history of flow, Dover used a simple capillary tube. What he did, he used a capillary tube with the blood inside it, a light source on one side and a detector on the other side. So as and when the cell was passing through the light source, an image was generated. Was a basic flow cytometer way back in 1934. After that, in 1949, Wallace H. Coulter came up with the Coulter counter or impedance-based method. So what is impedance? Impedance is when an object is suspended into the fluid medium, it will displace the volume of fluid according to buoyancy force. Okay? So that was the principle in the Coulter counter. Then in 1953, Crosland Taylor, he used the first laminar flow for optical counting of RBCs. In 1965, Max Fulweiler, he developed the first cell sorter. So cell sorters was basically to sort out different kind of cells. Wolfgang Bode, he used the fluorescence, the biggest fluorescent markers which are available in the market. So he did, and then that the history of flow cytometer, the development of flow cytometer was started way back in 1934, and gradually it has progressed to what we have today, the various color flow cytometers like 12 color, 14 color and up to 25 color flow cytometers we have based on the time. Yeah, it is on moving to the next. So what is a flow cytometer? Flow cytometer is a technique used to measure the physical and chemical properties of the cellular component, and cells are visualized based on their morphology and staining characteristics. So flow cytometry, that's analyzes the multiple physical characteristics of the particles, usually cells, as they flow in a fluid state, usually a laser. Size of the cell, it will help you in identifying the internal characteristics of the cell. It will help you in identifying the antigenic properties of the cell. So it tells us about the size with the help of the forward scatter, about the relative granularity or internal complexity of the cell with the help of the cell. So for the basic flow cytometer, we need 4 components. They are the fluidics through which the cells will flow. There are the optics, which will emit the lights. There are detectors, which are placed to detect the emitted light, and the electronic system. So these are the 4 basic components. This is the basic principle. You have a sheath reservoir with a sheath fluid inside it, and you have a sample tube. Now there is a tube through which the air pressure is injected. Now both these tanks are airtight containers. So when you inject the air pressure into it, the sheath fluid line. Similarly, because of the air pressure in this tube, the sample will get injected into the sample line because of the pressure. Both of these are airtight containers. Now this sheath fluid will flow from here and come into this flow cell. Similarly, the sample will come through the sample line and come into the flow. In the center of this, you have the sample. Okay. So, what is the function of the sheath fluid? If you imagine any kind of this kind of tube where a liquid is flowing, the center part will be at the highest speed. Okay. Because of the friction offered by the walls of this tube. So, with the help of this sheath fluid, we are trying to, because of the pressure generated with that, we are trying to make this sample into a single cell suspension, so that one goes ahead. Hydrodynamic focusing. This concept of using the sheath fluid or any fluid to make the center of the sample fluid into the single line because of the pressure exerted by it is called as the hydrodynamic focusing. The cell sample is injected into the stream of the sheep fluid by the flow principle. And cells in the sample are accelerated and individually passed through a laser beam for interrogation. So once you get a single cell pattern here, and suppose there is a light source here, so at a time through the light source only one cell will pass, and that is what we want. So there should be one cell at a time. Sample should be free from any kind of debris. Or any particle, they will also generate some kind of impulse. So while processing the fluid, you should be very careful that it is debris free. So this is just an example what happens if you change the pressure in the sample tube. If the sample at a time, but if you want to acquire the sample fast and increase the pressure or increase the intensity of acquisition to medium and high, then you will get multiple cells passing at a time. But this should not happen, because if more than one cell are passing through at one time, then it will be counted as one cell, or you get a different type of signal. So that will interfere with your data analysis. So it is always preferable that a single cell passes through the laser source. So ideal cell concentration in your sample that you have processed should be 1 to 2 million per ml, and the flow rate, low flow rate is 30 microliter per minute and high is 60 microliter per minute. This is the flow rate or the speed of acquisition of the sample. Coming to the optics, we have excitation optics and correction optics. The excitation optics are the source of light that we are using, and usually we prefer light, or the lenses of shape are used to shape or focus the laser beam to a particular cell. And then we have collection optics, which are the detectors and the filters. So I'll be telling you about these things in a short while. There are different types of optics. Here you have the neutral density filter. Now what is the function of the filter? What it does, it will allow only some amount of it to pass through. This is the function of the neutral density filter. Why do we use it? Suppose if the light source is very strong and it goes and falls on the detector directly, it is going to saturate the detector. So we place this neutral density filter in the area so that only the detector. Then we have a beam splitter. The function of the beam splitter is that suppose a ray of light is passing through it, it will allow only some amount of it to pass and the rest will be reflected at 90 degree. So for neutral density filter and beam, there is no wavelength specificity. The lights of all the wavelengths to pass through. Okay, and then you have long pass and short pass filter. So these long pass and short pass filter, based on what kind of filters they are. So these are the different kind of optics that you use. Coming to the light source, we can use various light sources which are available, like the arc lamp, the lasers, or the LEDs. But we prefer the laser light because of here is usually a. So we prefer to use this, and also because it gives the pronounced signal to noise ratio. That is, we can differentiate between the signal and noise to a larger extent. There are other light sources like arc lamp, but we don't prefer to use it because the light is non-coherent. Similarly, LEDs we don't, because it produces light of one wavelength and which is available in visible UV and infrared wavelengths, and moreover the light produced is non-coherent. So we prefer to use the laser light. Okay, so coming to the basic property, what is forward, what is side. Here, and this light is going in this direction in the path. In its path, if you get a cell here, what will happen? The light rays which are touching the sides of the cell will be refracted this way. The some amount of light which are going and hitting the internal structures like granules or nucleus, they will be reflected at various angles. So there are detectors placed all over at different angles. Rays which goes and hit a detector here will tell you about the size of the cell. Okay, so this is called as the forward scatter, tell you about the size of the cell. And those light rays which are reflected at various angles, they will be caught by the detectors placed at various. So they will tell you about the that is the granules of the cell, the lobulations of the nucleus. So the more granular or more lobulated a cell is, more will be the side scatter. And the larger the cell is, it will come far ahead on the forward scatter. So forward scatter, size of the cell, and side scatter is complexity or granularity of the cell. Okay, so this is how image is generated. Suppose this is a cell passing through the. Here, if the cell is small, you will get a small voltage pulse. If the cell is of medium size, and as the cell increases in size, the voltage pulse of the pulse increases. So this is how you. And about the internal complexity, internal granularity or lobulations of the cell on the side scatter. Now, if you would have heard PITC, PA, APC, these are the different fluorescent markers available in the market. Why do we need to use them? Suppose it is a cell or tissue, it has an antigen expression on its surface. Now this cell or this antigen is very small in size, you can directly visualize it. Antibody to it. But this antigen antibody, these are again very small to be visualized by microscope. So what we do, we tag this antibody with a fluorochrome. So when a light hits this fluorochrome, it will emit a light of specific wavelength depending on the kind of fluorochrome that we are using. So basically the flow cytometry is a fluorescence based technique. So we use the fluorescent, these fluorochromes or fluorescent markers of different types which are tagged to the antibody, and finally this antibody will go and attach to the antigen present on the cell surface on the marker. So the fluorescence is bestowed upon a cell through the use of fluorescent dye called fluorochromes. The laser beam excites the fluorochrome at a specific wavelength which is called as the absorption wavelength. And it emits the light at a different wavelength which is called as the emission wavelength. So you have absorption wavelength which is different. And this shows any light falling on the fluorochrome will excite the electrons inside it. This is called as the excitation, and once anything gets excited, the electrons get excited, they will emit the waves or emit the energy level. This is called as the emission wavelength. So light emitted upon decay of the excited molecule is called as the fluorescence. This is a list of the different kind of fluorochromes which are available in the market, and you can use them. So if it's a 4-color flow cytometer, say 8 color, you will be using 8 fluorochromes. Okay? So ये basic है. Depending on the color of cytometers, अभी बोलते हैं 10 color flow cytometer और 12 color flow cytometer. तो ये color क्या है? Color is nothing but the number of fluorochromes of different kinds that you can use to tag the antibodies. Okay, now one more thing, the antigen expression on the cell varies. Some cells will have many antigens attached, some will have only few antigens attached. So depending on the number of antigen attached to the cell, you will get the pulse. If there are only few antigens, you get a weak pulse. If there are many antigens, so depending on the antigen present or the number of antigen or number of the binding sites present on the cell, either you will get a weak signal or you will get a strong signal. So depending on the signal intensity, you can get to know the antigenic expression of the cell. Now these are the various band pass filter. Long pass filter is, suppose it is long pass 500. What does it mean? It will allow wavelengths of more than 500 nanometer to pass through, and those wavelengths which are smaller than 500 nanometers, जो बड़ी है stop, blocked or reflected back. Similarly, you have a short pass 500. Short pass का मतलब क्या है? The wavelengths which are smaller than 500 will pass through, and those longer will be reflected back. So these are the various kind of filters: long pass filter, short pass filter, and the band pass. Band pass filter means suppose this is 5 is wavelength of 450 to 550 will pass through. The others will be reflected back. So depending on your requirement, you can use the various kind of filters which are available. And this is again the graph showing, suppose this is long pass 500. So more than 500 wavelength, all can take it. This लाइव जाने के बाद क्या करना है कैसे ना एक्चुअली ये जो सिस्टम है ना यहाँ कली लगा है You have a lecture theatre downstairs, so usually for this kind of presentation, we use the lecture theatre. But screen का lecture को display screen, तो इसमें इसलिए हम लोगों ने arrangement किया है पर choose किया है. This is the basic model of the flow cytometer. ठीक है? This is the laser beam or the light source, and this is the object or a cell which is passing source. Here, so what happens? Whatever laser beams come and strikes this object, so some will come and strike this neutral density filter. So I told you the purpose of neutral density filter is that it will block some amount of light and allow only part of it to pass through, so the detector here doesn't get saturated. Nanometer, and the light which is coming out is also a 488 nanometer, the same wavelength. And this light gets caught by your band pass, it passes through the band pass filter 488 plus or minus 10, and then you have a photodetector here. So this source, this light which passes comes in the forward direction is your forward scatter, and it determines the size of the cell. Reflected, and here you have a long pass 510. Long pass 510 means more than 510 will pass through, the smaller one will get reflected from here. And here again you have a band pass filter of same radius plus or minus 10, and here you have photodetectors. The difference between photomultiplier and photodetectors I will be telling you. Here you have a photomultiplier tube. Now again the light which is passing through, here you have a 560 nanometer long pass. So more than 560 will pass, the less than 560 will get reflected back. Again you have a band pass 530. Now how do you decide 488 or 530? These are your different fluorochromes with different wavelength specificity. Like suppose this is a FITC channel. Here you can have the signals from 480 to up to 550. So whatever comes in this direction will be FITC channel. Similarly, 560 will be your, whatever passes through will be your PE channel, and then you have the final light passing through will come in your APC channel. So depending on the kind of filters you are placing, the amount, the wavelength of the light which is allowed to pass through and which is and caught by the photodetectors, or that will decide where you are going to get your signals. So this is the basic model of the. Now the difference between the photodiode and the photomultiplier tube, which I told you initially. The photodiodes have a quantum efficiency of more than 90%. Now what does this mean? This means wherever you have kept the photodiode and the light is falling on it, it will allow more than 90% of light to pass through, or it will reject more than 90% of the light. Whereas the photomultiplier tubes will detect only if it's a green channel, 25% of the light, and if it's a red channel, 15%. So we know that photodiodes are more stronger as compared to the photomultiplier tube. But if you remember the previous diagram, photodiodes forward scatter and the at various angles. साइड स्कैटर पे जो लाइट जा रही है वो एनीवेज वीक है, तो वीक लाइट जा रही है, फोटो मल्टीप्लायर ट्यूब भी कम इंटेंसिटी ऑफ एनर्जी अब्सॉर्ब कर रहा है, स्टिल वी हैव ट्यूब एट द साइड एंगल्स. There is something called as internal gain. Internal gain is more for the photomultiplier tube when compared to the photodiodes. What is the internal gain? Internal gain is as you go on increasing the voltage of the channel, the amount of light emitted or the signal emitted are more, so that thing is multiplier tube. So that is directly proportional. The amount of increase in the voltage is directly proportional to the wavelength of the signal intensity. Hence, we keep photomultiplier tube for the side scatter at various angles. Whereas the photodiodes have the very small internal gain. That means when you increase the voltage to certain wavelength emitted will increase, and then it will come to a plateau and it will stop increasing. So, what is the purpose of the photodetectors? It transforms the light into electric current. The types of photodetectors that you can use in the flow cytometer are the photodiodes or the photomultiplier tubes. This is the basic diagram of the wire tube. The anode. So when the light source strikes it, the electron will be generated. It will go and hit here, and every time it strikes with a dynode, it will multiply. You have 1, then 2, 4, 8, 16, 32. That way, the electric signal will, the electrons will bang, and you will get a stronger electric. So how is the pulse or signal created on the flow cytometer? This is a cell passing through a light source. So as the cell enters, you will get the initiation of this curve. When the cell reaches the center of this light, you will see this curve reaching to the peak, and as start coming down. So this is how a pulse is generated. On the y-axis, you have the intensity, that is the number of antigens being expressed, and on the x-axis, you have the time. There is something called as analog to digital conversion. I told you in the beginning there were balls of varying color kept in the basket and you wanted to separate them. So how do you do? Yes, this as I showed you in the previous graph, on the y-axis you have signal intensity, on the x-axis you have time. Now in analog to digital conversion, what happens? Your this is the signal intensity. Suppose there is 400 nanometer wavelength, then you have 800, then you have 1024, the wave of different wavelength signal intensity you have. And on the y-axis you have number of cells. So once a cell passes through this laser beam, which has 400 nanometer signal intensity attached to it, get a pulse here. If a cell with 800 nanometer passes through, you get a pulse here. So if you have two kind of cells, some with 400 nanometer intensity signal, some with 800, you will get the number of cells in different things. So now you know that how many cells had the signal intensity 100 nanometer wavelength and how many had. So this is how you separate the different kind of cells. What happens if you get two cells passing at the same time? In the beginning itself, we discussed that the cells should go in a single file pattern or one cell at a time. So if you get two cells passing through this kind of peak, and this is going to interfere with your data. Okay, this is called as doublets. So in this screen, now suppose two. Intensity of the signal, the height of the signal remains the same, that is the intensity, whereas the area increases, right? The area increases here because the two cells are which is coming in this linear pattern. So probably in the next few presentations we'll learn this. So once the data file has been saved, the data can be displayed in a number histograms. You can display the data in form of histograms or scattergram or density plots or contour plots or tomogram plots. So you have a different kind of presentation. cells or the events which you get on the x-axis are positive for FITC. Those you get on the y-axis are positive for PE. And here in this quadrant, the cells are negative for both the markers. Okay, so we'll tell you when we discuss about acute leukemia and further. And so you can change it to scattergram or control plot or histogram. And what do you mean by gating? Gating means choosing a population of interest. Suppose you want to study the lymphocytes, you put a curve or a gate around it. That is called as a gating. So this population, I'll put this gate around this marker and study further what it is expressing. So this is called as gating. Suppose I want to study these cells, if I put a gate around here and this population, then and this is a basic plot. Just wanted to tell you about this. This is a forward scatter and this is a side scatter. So forward scatter is for the size. So as you know, the lymphocytes are the smallest cell. So you will get the lymphocyte population here. And this will come and lie slightly above the lymphocyte and slightly forward. Similarly, the granulocytes, they have more of granules, more. Those will come slightly up or higher up on the side scatter. So this is a basic plot. With this, I will end my presentation here, and I would like to call Dr. Jasmita for the next presentation.
How many of you over there are able to see the screen? All good? Talk about instrument setting, antibody and panel selection. So these two lectures, basics, panel selection, they are the most boring lectures for flow cytometry workshop. Why? Because you want to see data, and you want to see cells, and you want to see plots. And what we are showing you is how it can be generated, how to do. So about the antibody selection panel. What do you mean when you talk about the panel? So how many of you are active flow cytometry users? So how many of your departments have a flow cytometer? Right? So a lot of it. But you don't get eventually. So a panel is, have you heard of types of an acute leukemia? So number of in how many tubes you're going to put there, and that depends on the flow cytometer that you have. Right? So if your flow cytometer has an 8 color capacity maximum, then your tube cannot be more than 8 markers. Okay. Or if you're trying to measure more markers than that, then also there is an option, but it is not utilized in acute leukemia diagnostics as frequently. So now if you have to make a diagnosis, distinguish between lymphoid, myeloid, and say the third one, BT and myeloid. So you have to have set of markers of each lineage. So 8 markers in one tube doesn't mean that you will have different markers in say the 4 tubes that you're going to put, because you want certain gating markers. So you heard gating. So for gating of normal peripheral bloods, marrows, or malignant marrows, peripheral blood, which is the marker that is most frequently used? CD45. So you want CD45 in all the tubes, right? Sometimes it becomes difficult to identify blasts, so you would like to have CD34 maybe in all your tubes. So that takes away 2 channels. Remaining 6 are there if you have a 8 color flow cytometer. So you have to adjust your markers accordingly. And now each of these markers doesn't have an equal expression on the cell. So some markers will be expressed, say if you have a cell, some markers will have 10 molecules, the other markers will have 100 molecules. So say CD34 has only 10 molecules in a cell. So what do you want to target with a brighter fluorochrome so that it is able to pick up those 10 and resolve it from the noise? However, if you have something like CD45, you just need it for gating most of the time. So for gating, it doesn't matter even if it is on a weaker fluorochrome, because it is still able to resolve your populations. The idea is to resolve different populations. So that is how, when we talk about a panel, that means that it is a combination of antibodies tagged to various fluorochromes that you're going to multiplex in a single tube. And eventually, a panel can have a single tube, it can have multiple tubes. Can you give me an example of a single tube panel? So when we do PNH testing, you have two tubes, you have WBC, and if you're doing RBC, then the RBC tube, right? Other than that, you don't need much. So if you're resolving, say, an assay for chronic granulomatous disease, or no, not chronic granulomatous, let's take an example of leukocyte addition defect. So in that case, also you can have just one tube and it will give you the entire resolution. So it depends upon what question you have, what equipment you have. Now, say somebody has a 12 color equipment. And if you are doing an acute leukemia analysis, maybe the 5 tubes of an acute leukemia in an 8 color setting can be fitted into 3 tubes in a 12 color setting. So it also depends on the number of colors that your equipment has. So fluorochrome you've already covered, will absorb at one wavelength, will emit at a higher wavelength. Okay. So when you have a look at the fluorochromes, the conventional ones, FITC, PE, they are relatively smaller molecules. FITC is a small molecule. Among the large molecules, you have PE and APC. Now, this has an important connotation. Why? Because when you have to permeabilize the cell, something in the intracellular fashion, and if you have it tagged to APC molecule, then your permeabilizing time becomes critical. It has to be long enough so that your large molecule APC can enter the cell. And trust me, we faced this challenge recently when we moved the CD3 from surface to cytoplasmic in APC. So in the APC channel, the regular permeabilizing time of 8.5 minutes, which was working very well for us, was not working anymore because CD3 was tagged to a large molecule. So we had to increase the permeabilizing time to up to 12 minutes to ensure that all the CD3 goes in. So this is just an example of a large size one. Then there are tandem conjugates. So tandem conjugate means you have one fluorochrome, say PE, that absorbs the energy. You have a Psi7 molecule attached to it. So now the energy emitted by PE excites the Psi7 and Psi7 emits energy. Advantage that if PE was detecting at a certain wavelength, by giving it to Psi7, the wavelength of detection moves ahead. So you can add more dyes together. So that is the advantage of tandem dyes. And what is this? Emitting light always has lower energy. So whatever you excited with, the emitting light will be of a lower energy, which means that it will move ahead in the spectrum. So if you have the excitation light is a blue laser, the first emitted light is FITC, that is in the green area, PE is in the orange area. So it will keep on moving ahead. And this difference between the emission and the absorption light is called as Stone's Throw. Why are these terms important? Because you need to know where your fluorochromes are going to emit light. So whenever we are designing a panel or selecting antibodies, we have to first decide what is the question that we want to address. So if the question is clear, then only we will know what markers have to be put, what is our experiment goal. So you just gave me an example for an acute. You want CD45, you want site scatter, you want immaturity markers. Then you have surface and cytoplasmic markers that you need to use. Then the next is the biology. You have to understand how the antigens are expressed. What antigen is at a high intensity, what antigen is at a dimmer intensity. In addition to that, you have to know your flow cytometer, and I just gave you examples, 8 color, 12 color. Then sometimes, we frequently talk about blue, red and violet lasers because these are the most frequent equipments available. But there are also equipments which instead of the red laser will have a yellow-green laser or will have another laser. So with each laser, the excitation of the same fluorochrome and the emission may not be the same. So you have to understand which fluorochromes work on which particular equipment. The next question is, you have to maximize resolution. What is resolution? The difference between negative and positive signal. So you want your positive signal to be as far away from the negative signal as possible, right? You don't want them overlapping when you're gating. Otherwise, you may be getting junk and calling it positive. So that is why fluorochromes are matched to brightness. The example that I gave you of 10 molecules over a surface versus 100 molecules over a surface. So anything which has 100 molecules, you can use a dimmer antigen, for example, and anything which has 10 molecules, you will need a brighter fluorochrome. And along with that, you have to first review the theoretical panel design. So sometimes panel design is also dependent on which particular antibody is available with which fluorochrome conjugate. आपको चाहिए कि आपको से PV605 के साथ में आपको CD81 मिल जाए, but it may not be so that all the manufacturers have thought of the same thing and they are manufacturing 81 with PV605. So there are certain critical antibodies with which, or newer antibodies with which the number of fluorochromes they are conjugated with may be limited. So you may have to change those and revert to a common antibody conjugated with these rarer or uncommon fluorochromes. So first is reviewing your theoretical panel design, and the next step is to optimizing a panel. अभी आपने पैनल बनाया, जरूरी नहीं है कि 100% of the time it will give you correct information. So sometimes what happens is that we make a panel, we try it in the lab and we realize, oh, this antibody is not working. Right? So in that case, you have to then change that antibody or modify the fluorochromes for one of the other antibodies. And why have I drawn these arrows? Because at every step, you may have to go back to the previous step. You may have to do that step all over again. So I'll give you another example. So we had the Cyto 3 APC shift that I was. So Cyto 3 initially was with PAC group. So we shifted 7 to PAC group. And we realized that 7 for PAC group works well in our acute leukemias, but when it comes to our MRD analysis of ALL, then the 7 PAC group is not able to separate the positive from negative completely. So it is still separating it, but there is an overlap. So we are taking a lot of junk in the 7 positive area because we don't have two clear separated populations. So because of that, now we have shifted 7 to BB421, which is a brighter diamond PAC. So this is what I'm trying to tell you, that it will never happen that the panel you design will work 100% functionally well in the first loop. Which is why it is important to review the existing published literature. Sometimes adopting somebody else's well-functioning panel is better rather than having to make your own panel and struggle with it. And this process that I've shown you is called as iteration. So knowing your population of interest is important because you want to know what your panel is going to answer. So what is your panel's objective? What are the populations that you're going to get? What markers are those populations going to express? Now the choice of tissue becomes very, very critical. If somebody wants to do lymph node flow cytometry, lymph node is a solid tissue, you have to convert it to a single cell suspension. For that you have to mince the tissue, break it, pass it through seeds so that you get single cells. So that is the difference between, say, flowing a liquid tissue like peripheral blood or marrow vis-a-vis a solid tissue. Then intracellular and surface markers are very, very important because some markers you want to assess on the surface, other markers are intracellular. Obviously, for intracellular markers, you will have to drill holes in the cell. Similarly, someone of you may want to assess cytokines tomorrow. You may want to stimulate your cells and assess cytokines. So those are again issues that come up. Small issues in which people who are working with flow cytometry are familiar with this. Sometimes you have a set of antibodies available and you have a short-term change that you want to get. So right now you can't order the next batch of antibodies, but you want to optimize using your existing antibodies. So the goal, whether it is short-term, long-term, are all important considerations here. So how do you select markers? You first have to know how many markers you want to answer the question. For example, for an acute leukemia diagnosis, you have 45, you have 34, then you want CD19 in your panel to identify B lineage, you want cytoplasmic 79A, at least in the first row itself. Then you want Cyto 3, surface 3 maybe. Then you want the myeloid markers, and there is a whole list of these markers. So therefore, you want to know how many markers are required to answer the question that you have, which populations you want to record. So even if you want to record blasts, that does not mean that you will leave every other population behind because they are your valuable internal positive and negative cut codes. So now you have different markers and I will tell you what are primary, secondary and tertiary antigens. A very, very important factor is which markers are co-expressed on the same cell marker. For example, when we talk about B cells, CD19, CD20, they are all co-expressed on the B cells. Right? For CD4 and CD8, they are co-expressed on T cells. So you have 4 and 3 and you have 8 and 3. So in that time point, marker co-expression is important. And you have to think of the gating strategy when you are deciding the panel. So gating does not come and the gating strategy doesn't come after you've acquired the data. You have to have a flow of thought in your mind. Okay, if I have this panel, I will use this marker for gating purpose. Then I will subgate using this marker. So that clarity should be there in your mind first. And the choice of clones is important. So there are many antibodies, you have an antigen. In this molecule itself, there are multiple epitopes which are used to make antibodies against which the antibody is targeted. So choice of the clone, very, very important. It's for each individual vendor also your the antibody. It will give you SCS log. You can open it, the technical spec sheet, and you can see what is the clone, how is it reacting with lymphocytes, with plants and everything, the data that they have provided. For markers, you have primary markers. Now, these are lineage defining markers, usually expressed at higher intensity. So, for example, for a T cell, CD3, for a B cell, CD19. These are primary, and they as easily as possible. So these markers, relatively dimmer fluorochromes also are okay because they are expressed in populations that are in normal populations that are high. It doesn't mean that on malignant populations they will be at the same intensity. Population they are expressed at high. Are relatively better characterized as in there is more experience available with it. Simultaneously, they are intensities. Examples are CD20. CD4 and 8, 4 positive T cells, distinct single population. 8 positive T cells, you have a bright population, but then you have to the negative 8. Then tertiary are antibodies or markers that have a very low level expression on the cell. And these are sometimes critical in an assay. For example, for a T regulatory assay, FOX, PP, CD25 are these markers. So you have very, very dim expression of these markers. So anything that is something where you have a tertiary level expression and a brighter fluorochrome to say negative population. Now, clone decision and gating strategy, I have already told you that you can use your experience or the literature and so these are three tools. Fluorobinder is one. You can just register your account. Okay, how many vendors, for example, for the same antibody. So Fluorobinder does that with you. You input a marker, you input the clone you want, and it gives you apply that antibody. This is something where I struggled with. So we were using an assay for MDS, and we were using one of the clones for CD16, which was NKP50. And if you look at the myeloid cells for 13 versus 11B, which is this middle plot, you clearly see that the marker expression is normal, but we don't have any expression of 16. Normal internal 16. So NK cells were 16 positive, but somehow the granulocytes were not reacting with 16 at all. So we moved to a different clone example, and you see the way the 16 has reacted in the granulocytes. So this is an example of 16 working very well in lymphocytes, for which it was initially added. But what about the myeloid cells? It was not binding to the myeloid. So we had to go back, revise, and then. And this was an example of a patient of MDS. Now the next component is to know your flow cytometer. What lasers your flow cytometer has, what detectors are available, number of detectors per laser. For example, a Beckman-Coulter equipment on a blue laser will give you 5 colors, while a BD equipment on a blue laser gives you 4 colors. A separate channel called manufacturers, lasers, and detector combinations available. So these are examples. Again, this is from published literature. This is from every vendor's website that you can get details of their equipment. So down is the TXplex and on the top is whether which marker will say which BD antibody will work on a Beckman equipment also by looking at the emission and the now how do you select fluorochromes? So what we want is that all the markers that are available at different expression levels should be well from each other. And availability for the detector availability on our equipment guides the fluorochrome selection. But at the same time, pairing of antigens with respect to antigen density is important. So we've already talked about dim antigens on bright dyes, bright antigens on dim dyes, and co-express markers. We have to think about what the spread of the other marker is. So you have the emission for FITC, you have the emission for P, and then you have P, Texas, and together. The same way part, and most of the emission overlap. So you cannot multi-laser. The same example is there. 647. So this knowledge is important so that you don't end up with a panel where you have APC, where you are expecting to tag them to two different thickness. For the violet laser also, if you look at BV421 and B450, again the emission, it overlaps significantly. So these two dyes cannot be used together. Titrate antibodies. So why do we titrate antibodies? Not what you read here. Practically, everybody wants to titrate antibodies so that if something goes in, you save all the money. These are expensive things. But the reality of why is that we don't want our negative signal to be brought. Where we can resolve a positive, bright positive is okay, but a dim positive, we have less space to resolve it. Titrated to minimize noise. Controls are a very important thing is to prepare antibody cocktails. So if you have 8 antibodies in a tube, it is better to surface. For 10 tests, fixed amount of your cocktail. So every time you avoid pipetting errors if you're using cocktails, but you have to validate binding together is working in your cocktail as well. And you again have to make adjustments. So a bright fluorochrome, why is it called bright? Because it emits more fluorochromes. So for example, P is a bright dye. If you look at FITC versus P, the number of photons that have emitted for P, they are larger than the number of photons. And how do you assess this brightness in test conditions? You want to evaluate something called as a stain index. So every time you get a new, when you're doing the panel, you run a titration experiment, assess different concentrations of that antibody. Say if it is a antibody that is for a 5 microliter thing. So you can assess 5, 2.5, 1.25 and so on, or you can assess 5, 4, 3, 2, 1 and assess what is the stain index. So you have the red population, you have this blue which is your noise. So if you see the picture on the left, the noise in the signal is very low, while here but when you look at the particular population, so the area that it is occupying is far more. So this is what you want to optimize for, and you calculate this as a the one that you want is difference of MFI of negative from positive divided by 2 times the robust ST or the width of the negative. 0711 on the violet laser. On the blue laser, you have very bright dyes which are now changed to P. So this and the Beckman flow cytometer also, you know which dyes are brighter. Now the antigen density. You want to know what is the density of each antigen on a cell? Harness the published literature. For common antigens, you will have antigen densities which are published, and antigen densities that have been done on multiple flow cytometers. For example, these are examples of overlapping, but they are not exactly the same. So part of it is also dependent on the equipment. For co-expression, if you have 2 markers that are co-expressed, you don't have to worry over into the second channel. However, if they are co-expressed, then you have to look at what is the relative intensity of these markers. So between A and B, if A is much more than B, then what you need to do is use a bright marker for B. Why? Because it's a dim one, so you want to use a bright marker. The dimmer, it should not be that the A positive population, B negative population should not be separated from the negative. So that is a simple key that you use. Matrices for, and here you have areas marked in red where you have a lot of difficulty with co-expression. So if you want to say assess a marker that is co-expressed, you don't tag it in the adjacent channels. For example, here you can see that P is I7 versus BV786. Percentage problem. Use 2 common express markers on each channel. There are other things that affect resolution also besides the signal noise ratio. It is how you fix the cell, how cells are permeabilized. Permeabilized cells you and what the buffers are in your solution. There are additional considerations whenever you are doing the intracellular one, and then you plot something called as strain index and signal noise ratio. Signal noise is the MFI of the positive divided by the negative. So the area where it gives you the best fit of both, which is this 3 microliter as the final concentration of the antibody. And if you see the CD3 here, when titrating, we use the permeabilizing step so that we have the permeability be done for this marker. And again, here 2.5 was determined as the best volume. So in your panel design, these are the things that we talked about. You have to know what you are running, whether it is a simple, complex, long or short-term panel, whether it is surface, cytoplasmic or nuclear, whether your cells are going to be stimulated or not. You have to understand the biology of your cells, know your flow cytometer, and effectively maximize resolution. Again, testing setting. So this is the lyric. You will see that every instrument has a basic QC that has to be done every day. So for a BD flow cytometer, that is CS centigrades. For Beckman, it is something else. For any other equipment, it will be something else. Now, once you get an equipment the first time, you have to set up your instrument gains or what measures relative cell size. Relative means that cells in the same way, side scatter assesses granularity. So technically, you have to set the FSC, SSC voltages so that your populations are separated. You have lymphocytes, you had monos, you had granulocytes. You want them to be separated. You don't want them squished or scrunched together. Simultaneously, not working, if your cells are dying off, if there's a lot of maybe add BSA to your buffers. So bovine serum albumin helps the cells stay stable. You can increase the threshold on forward scatter. Say if you're acquiring at 10,000, you can increase it to 25,000, which means that till 25,000, the cytometer lets everything at all. Or you can increase the forward scatter voltage. Now, increasing voltage, you don't want your cells to go off scale on the plot. So there is only a certain amount to which you can increase. And to exclude doublets, enable not just the area which is the default, but also the height and the width for your scatter voltage constant, which means that when you're running your compensation experiment, at that time run a unstained sample also, so that you know which particular voltage is working well for your sample. So ideally, you should do your sample voltage. If say a lab is doing lymph nodes also, it is doing something like bone marrows also, then you can have different forward and side scatters for those, keep them stable in that particular assay. Do not randomly change these settings, and for the detector voltages, they should not be changed. So don't touch the FITC voltage, P voltage. Say, let me increase the FITC voltage to try and separate them. The separation will increase, but it will not be the only thing that happens. The signal for PE that spills into, the signal for FITC that spills into PE, into per CP, that will also increase, and you will not be compensating for it on that particular day. So this is a voltation experiment. Any given marker, same tube, low voltage, the separation is bad. High voltage, separation becomes very good. But what increases? The negative here, the spread here, that also increases. So you want an optimal voltage where the spread of the negative is low, but your positive population. Like for example, here 450 is the better voltage. Again, for most manufacturers, you don't have to do this, but a voltage change may be required only and only if your brightest population is off scale. So for example, CD38 on plasma cells is very, very bright. So that is the easy way to correct this, change 38 to a dimmer fluorochrome and you will have the solution. But if you cannot at that time point, what you can do is try to reduce the voltage of APC a little, do the compensation experiment with that voltage so that that assay works well. You should never do employee in this narrow range, P in this narrow range, but you have a spillover. So P continues to emit till about 650. So it enters into the P channel also. So P till P should stay. So constant control, a cell which is marked only with FITC. And if you see, the signal is in FITC, but it is also spilling into PE. So you are compensating to try to get the signal back into the FITC channel. And this is what is done here. And how do you do it? The negative population for FITC and the positive population for FITC, their intensity for PE should be nearly constant. And this should not be something which we do by eyeballing. This is something for which statistics should be used. And you subtract the FITC signal into in the PE channel. So this is how compensation will impact it. If you have a correctly compensated data, overcompensated, you get P cells. Undercompensated, you have a lower estimation, but the correct one is say 10%. So these are the sources of spillover. So adjacent detectors, FITC PE combination. So PE Psi5, this is similar excitation spectra. So cross laser, for example. So for this, many of the equipments have a laser delay incorporated. So blue laser light comes a little before red laser. So therefore they separate these two lights hitting the cell, and residual base fluorescence. So we talked about tandem lights. These are dominant emission will happen in the PE area also. So these are problems that lead to spillovers. Compensation controls, you can run single stain beads or single stain cells, and many of the manufacturers, they give you auto compensation also with their defined bead cells. So Lyric has a defined compensation beads which have to be approach or the single commercial beads or single stain cells. However, even if you do a good compensation experiment on your equipment, which is what most of us do, there are still small adjustments that are required, and they can be done after that also. So these are the rules for compensation. Positive population and the autofluorescence should be the same. Right? So you cannot say sides or neutrophils as a negative. That will not work. And controls should have sufficient events in both the positive and negative population. So at least 100 events in both, or more than 5% of the parent population is required. However, it does not correct for spread. So for example, is spreading. So you have something which is going into the percipity is going into the P psi 5, P psi 7, and this is what is called as. So when you have a single stain marker here, the separation is very good. However, when you have a marker stained with B, you have. So this is in my panel, for example, between CD19 and 34. So 34, if it is expressed on glass, that population of it hits 19. So I know that positive. So you get familiar with it over a period of time. I'll skip this, and these are the controls. Out of this instrument is mandatory. Assay control, autofluorescence are mandatory, and this you have in all your tubes. You have the unstained tube in all your tubes. Com controls, you run it monthly, 2 monthly, depending on your manufacturer's instructions. There are something called as FMO controls, which can be done if required. So I'll skip all these slides. So this is an example. If you see this 34, what do you feel? Positive or negative or dim positive? 34 here. See, this is a workshop. We are going to ask you questions. So, iffy, right? Maybe something in red is higher. Maybe you can see the background signal. So in P, in FITC, in PERSI, there's a lot of background that these cells are flowing. And this is the high autofluorescence of promyelocytes. So we have to have the unstained tube at times to help us. And I'll end there. These are my acknowledgements. I will thank Dr. Parekh learning through, and even subsequently he's been teaching us all this. My colleagues Ganesh and Richa and Kimil, who helps me out with the collection of this. So I'm happy to take questions if you have any, although I think you are all bored out of your rooms by now. So how many of you enjoyed a good nap? 2-minute nap? Thank you.
So mine will be a very short class, clinical application of flow cytometry, because we will learn furthermore. है ना? ठीक है? So, like what you know, what will be understanding in 2 days, like आज के जो workshop में acute leukemia और CLP. ठीक है? But there are other uses. So flow cytometry provides rapid analysis of multiple characteristics of single cells. जैसे हमारा 8 कलर फ्लो हम एक ही सेल पे एक साथ 8 बार करते हैं, ठीक है? तो एट ए टाइम एट ए सिंगल सेल पे हम 8 चीजें एसेस कर सकते हैं, है ना? ओके. In a single file, as told by Dr. Rashmi, ठीक है? The information obtained is both qualitative and quantitative. Qualitative, you mean by जैसे हमें कीमोथेरेपी में हमें केवल फिनोटाइप्स, CLPD में हमें केवल फिनोटाइप्स, पर जब हम CD34 काउंट करते हैं तो हमें ट्रांसप्लांट के लिए एक्चुअली वो 34 नंबर्स है ना. सिमिलरली जब हम CD19 कर रहे हैं या 20 कर रहे हैं तो हमें रेटिक्सिमल थेरेपी के लिए हमें कट ऑफ काउंट चाहिए, तो दैट बिकम्स योर क्वांटिटेटिव. PNH में यू नीड अ नंबर, है ना, quantitative. For immunophenotyping a variety of specimen, you can use blood, you can use bone marrow, anything which can be brought into a liquid form in a single cell form. I know. So characteristic that can be measured includes cell size, cytoplasmic range of membrane bound or intracellular proteins. Okay. So one by one, most common is immunophenotype. Okay. ये एक like your morphology slide. Can you say कि ये myeloid है, lymphoid है? नहीं. So you need flow cytometry. You need immunophenotype. Not exactly flow, लेकिन immunophenotyping आपको चाहिए. ठीक है? Whatever. Okay. So by this है 33 है, चलिए DR है, MPU है, so it is my myeloid. ठीक है? But I can't see any granules or rod in morphology. So here helps my flow cytometry. So the most common is immunophenotype. Myeloid blast crisis है, lymphoid blast crisis है, CLPD diagnosis है, then clonality assignment. जैसे plasma cells. How do I know they are polyclonal or not? By morphology I can't. ठीक है? Minimal residual disease. ठीक है? Characteristic that particular like B cell का myeloid है 1333 MPU. Okay. Quantitative analysis. ये क्या दिख रहा है? I don't expect actually. This is your CD34 analysis, which we do for any. पहले you need this number. You need this number. ठीक है? How many bags should be collected for transplant will depend on this number. ठीक है? So this is my for enumeration. That is a quantitative analysis. ठीक है? मुझे number चाहिए, है ना? There is certain number जिसके बेसिस पे मैं बैग्स I'll stop basis, है ना? Then what is this? तो पढ़ पा रहे हो ना? बोलो ना, 4-8 एनालिसिस कर रहे हैं ना, 4-8 सेग्रीगेशन कर रहे हैं. Why do I need this 4-8? इसमें चाहिए होता है. हाँ? इसमें most चाहिए ना CD4 काउंट. ठीक है? तो होल लाइक जो आपकी मॉनिटरिंग करते हैं CD4 काउंट पे करते हैं ना. हाउ डू यू नो दिस? ना इसको काउंट कैसे करते हैं? बाय फ्लो साइटोमेट्री. ठीक है? ओके. इधर क्या है? इधर क्या है? So this is basically CD19 क्वांटिटेटिव हम करना चाह रहे हैं, ठीक है। मैंने यहाँ से फॉर्म स्कैटर्ड से लिम्फो उठाए अपने 3 और 18 पे मैंने सेपरेट किया, क्या needed in cases of rituximab therapy, ठीक है, because there are 20 specific for 20, ठीक है। In certain diseases जैसे certain immune disorder, certain neurological disorder, we actually need that cut off चाहिए होता है, ठीक है। और 19 का नंबर चाहिए, so that's again a quantitative analysis, ठीक है। अदरवाइज क्वालिटेटिव आप करते हो, सीएलपीडी में भी करते हो, ल्यूकेमिया में भी आप 19 लगाते हो, ठीक है। बट यहाँ पे जो आपका क्या कहना चाहिए, जो मकसद है दैट इज योर नंबर, ठीक है। तो दैट ओके। नेक्स्ट कम्स योर इरिथ्रोसाइट एनालिसिस। एनीबॉडी, क्या है ये, क्या दिख रहा है यहाँ, 59 235 पीएनएच, ठीक है। सो पीएनएच में दिस इज अगर इसको कैटेगराइज करोगे इरिथ्रोसाइट के एनालिसिस में, तो अगेन इट इज अ डिसाइड बाय फ्लो साइकिल। अर्लियर used to be done by card test, gel card test, ठीक है, ओके, बट इट इज बेटर बाय फ्लो, ठीक है। इरिथ्रोसाइट में करने से हो जाता है, पीएनएच में होता है, नहीं होता है, और नहीं होता तो क्यों नहीं होता। pH is a hemolytic anemia. Okay, so pH is a hemolytic anemia, so that is will, ठीक है। And if there is bleeding, hemolysis हो रही है तो flow कम हो जाएगा, ठीक है। So you assess pH by other two lineages, क्या है वो, monocyte and neutrophil, by two gating marker and two. So that is being shown over here. Okay, that is just a ये क्या है, ये क्या है, रेड क्या है, रेटिकुलोसाइट्स है, ठीक है। द मोर इमेच्योर रेटिकुलोसाइट, द मोर विल बी आरएनए कंटेंट, डीएनए कंटेंट, व्हाटएवर, ठीक है, आरएनए कंटेंट होगा ना, सो दे विल हैव मोर फ्लोरोसेंस। तो वी आर यूटिलाइजिंग फ्लोरोसेटिक these, what is your HFR is the more immature kind of reticulocytes, ठीक है, and that is your mature RBC which do not have fluorescence, ठीक है। So you can actually separate on the basis of both these RBCs. This is your system, है ना, है ना। आपके सिस्मेक्स में भी जो रेटिक काउंट आता है वो इसी चैनल से आता है, ठीक है, ओके। ये क्या है, इस पे कुछ लिखा है ऐसा, नहीं बता पाओगे। दिस इज ऑस्मोटिक, दिस इज अदरवाइज बाय योर स्पेक्ट्रोस्कोपी, ठीक है, बट यू कैन असेस बाय फ्लो साइटोमेट्री, ओके। प्लेटलेट एनालिसिस, ओके। वेल इक्विप्ड, क्या चाहिए, पीछे रखा है, अगर देखा होगा तो पहचान जाओगे, क्या है, एग्रीगोमीटर, but you can assess by flow 4161 in plants, absence of 42 in one can be assessed by flow cycle, ठीक है। and it do not have that limitation जो आपका एग्रीगोमीटर has a limitation, क्या लिमिटेशन है, 1 लाख काउंट होना चाहिए आपके प्लेटलेट, ठीक है। So you that. Next use, transfusion medicine. So what happened in your RH diseases, there is P2 level. Okay, so you can actually detect that fetal hemoglobin in matter by cell cycle analysis. Okay. So DNA content measurement. In cases of, it requires use of DNA interactive. What it does, these are different dyes like DAPI, PI. What it does is it directly correlate with the amount of DNA in the nucleus. Okay, the fluorescence of this dye will directly correlate with the DNA content of the cell. Okay. Ploidy analysis. In cases of so this is actually again based on the DNA content. This is the DNA content. Ploidy. Okay. Other use. Apoptosis. है ना? मार्कर लगाते जाइए कुछ भी एसेस करता है. Okay. 780 AAD is a viability mark. है ना? Means जितना 780 बढ़ता जाएगा, it means उतना आपके सेल कम वायबल. है ना? So annexin 5 your cells are viable or not. Apoptosis is there in research. Cell sorting. Sorting में क्या? Separate. We can actually separate those cells by electromagnetic separation. ठीक है? तो मुझे सिर्फ T चाहिए, CD3 cells मैं अलग से separate, उसको मुझे आगे stimulate. ठीक है? So I can actually separate my CD3 cells. This is cell. And then ये क्या? हाँ? कौन से वाला? ये वाला स्टूडेंट. ये मेन्यू ये क्या ये हो गया? This is okay. And see the use of that drug by various pharmacologic studies. Yeah. Tumor है कि नहीं? Pleural fluid metastatic है in those cases. Okay. So to summarize, this is actually what I have told you earlier. This is a table of. That's all from my side. So now we have a basic approach how to approach a case of acute leukemia. So firstly, what whole of flow cytometry morphology, we can identify blast, we tell that it's an acute leukemia, then by flow. First is the flow also helps you to detect blast. The next is it helps you to assign the lineage, whether they are lymphoid blast, in that whether they are B lymphoid or T lymphoid, then in the myeloid lineage, whether they are subsequently differentiated into erythroid, megakaryocytic, monocytic, which lineage is that. Then it helps you to identify the maturation stages and you can classify your BLL, TLL and myeloid leukemias further. Then additionally, now we know that WHO has made mandatory cytogenetics for our acute leukemias. So does flow provide us any clues for certain genetic subgroups? Then obviously you can identify some leukemia associated immunophenotypes which might help in the subsequent MRD detection. And lastly, the markers are being employed for use as targeted therapy. So now the standard approach for a case of acute leukemia is follow the Euroflow panel. In this panel, they have planned the immunophenotypic analysis in 2 steps. First is an A-lot, that is the acute leukemia orientation tube. I'll tell you the marker subsequently. Once you get the results of this A-lot tube, then you proceed to complementary panel, which is for B-cell precursor ALL, TELL, or the AML or MDS panel. So this is just a flowchart of how they say if you have a high suspicion of acute leukemia. Example, you have seen the blast cells, or if you have unexplained cytopenia, or you have sustained monocytosis, unexplained eosinophilia, then for all those cases you have to run a A-lot tube, and depending upon the results, you can go for a B-cell precursor ALL tube, which helps you to classify various subtypes of B-cell precursor ALL, along with evaluation for certain leukemia-associated immunophenotypic markers. If you get a result of TLL, further subtyping of TLL and diagnosing the ETPLL. Then for the AML MDS, you can classify it as various subtypes of AML. So this is again from the Euroflow panel. This is their 8 marker A-lot tube. Now you can see for the here what they have used is the gating marker is CD45. It also helps you tell that whether there are immature cells which will fall in the blast region. Then the immaturity marker here they have taken CD34. Now we know that there are other immaturity markers as well, TDT117, but they have preferred CD34 over TDT because it is a surface marker. The antigens are there on the surface of the blast. You need not use a TDT because of there's a nuclear marker. Then for the B lineage, they have chosen CD19 because it is a pan T cell marker, but it is not specific. We often see reports of AML expressing CD19 as aberrant marker. So they have chosen again another marker, cytoplasmic CD79A. This is a cytoplasmic marker. Another cytoplasmic marker which they tested was CD22. But they have preferred CD79A because again it is specific as compared to CD22. Then for the myeloid lineage, they have kept cytoplasmic MP as the marker. Now, when they were planning these tubes, they wanted a marker which can detect that particular lineage throughout the maturation stages, as well as it should have been specific. So for myeloid, it was just MPO which could fit into this criteria throughout the maturation stages and specific. The other commoner markers like 13, 33, they are frequently observed aberrantly in other lymphoid leukemias. Then for the T lineage, they have kept cytoplasmic CD3 and surface CD3 as well. Now when you go and read the maturation of T cells, जैसे T cells mature होते हैं वो CD3 surface पे आ जाता है. So they have kept these two, cytoplasmic as well as surface. If there is only cytoplasmic, then it means it is an immature cell. If it is having both cytoplasmic and surface, then it means that it is a mature T cell. So now these markers which I did have markers for each lineage. What do you mean by backward markers? कि अगर हमारा initial BLL हमें लग रहा है ये लॉट करने के बाद, तो जो आगे की आएंगे उन सारी ट्यूब में ये तीनों मार्कर 45T. So we identify the blast using all these 3 markers, and then we evaluate the subsequent markers in that. So that is backward markers will be all the tubes for further processing of BCPLL. Similarly, for TL, all the tubes will have 45 cytoplasmic CD3 and surface CD3. For AML, 34 and 45 will be there in all. So now coming to again, वो तो A lot tube है, but what the WHO says is how do you define a B lineage? Strong CD19, 79, 8, 22 or 10. Or if your CD19 is B, what do we mean by B? If the intensity is less than the normal precursors in 50% of the cells, then it is weak CD19. So then you need more than 2 markers, and that too will be strongly expressed. Myeloid lineage, again, as I told you, MPO is the one which helps you either in the form of flow cytometry or do you do it in cytochemistry or IHC. Or you can go for markers of monocytic differentiation, and at least 2 of them should be positive. That is NSE, CD1164 or lysozyme. The T lineage, it is cytoplasmic CD3 and that too should be strong. Or you can go for surface CD3 expression. Now for CD3, the antibody clone should be the epsilon chain antibody, not the other clones. You must take care of this. So that was part of a theory. Now coming to some plots. This is a dot plot because you can see all the cells here are represented. All the dots in this plot represent individual cells. So this is a basic plot of forward scatter. Now here you can see there are cells. Here you can see this is a blue population. There is a pink population sitting on this. So this blue has minimal. This is the population of your mature lymphocytes. Then you can see there are some cells which have some side scatter and they are a bit larger than this. This is your monocytic cells. And then comes these cells which are gradually increasing the side scatter. So these are neutrophils. And these are the cells which have the highest side scatter, eosinophils. Plot, if you have a normal sample, you will see this maturation pattern, these types of cells in the plot. And sometimes, but what we deal in acute leukemia flow cytometry, you won't have these differentiating cells. So what we need is a marker that is 45, which helps you identify the blast separately. So here again, you can see these similar 45 with a low side scatter, and again granulocytes are having a higher side scatter, and eosinophils are having the maximum side scatter, and all of these are having bright 45. So this is a dot plot which I showed you. Maybe go through the literature. Like you can have, this is a contour plot, you can see these rings. Then you can have this representation in the form of density, that is a density plot. So now in the A lot tube, I told you there are certain markers which are surface, that is they'll go and the antibody will attach to the antigen on the surface. Certain markers which I've seen TDT in IHC, वो nucleus में आता है. If you see MPO, it comes into the cytoplasm. So in the flow cytometry, we have to analyze these antigens which are inside the cell. So what we do, we permeabilize the membrane and we allow the reagent to enter inside so the antigen will bind with the antibody. So what happens is that changes the side scatter. Now you can see this is 45 versus side scatter for a surface staining of the same sample. So you can see the blast are here, the grans are reaching up to the side scatter of 200 and the lymphocytes are here. But once you have permeabilized the sample, what you see is these grans have fallen down because the cells shrink because of the permeabilization. So analyzing the plots, whether it is a surface tube or a cytoplasmic tube. Because what happens is, हमने यहाँ gate लगाया है, अब इसमें granulocytes भी आके गिर जाएंगे. So we have to take care that our blast gate is neat and clean. I'll show you more of it. So in acute leukemia, I told you कि वो forward scatter, side scatter से we cannot identify a blast. What we use to identify blast is CD45 versus, and these are your granulocytes. So your blast can have a variable expression. The most common is this pattern, that is they are dim. Lymphocytes से थोड़ा पीछे आएंगे और dim होगा. But they can have the expression. They can have this expression which can be made. They can even go to that is negative. So you say कि blast है ही नहीं sample में. we'll see more of it. This is a monocyte था, monocyte था, granulocyte था, यहाँ पे कुछ भी नहीं था. Now this region is the blast region. What you can have your blast, you can sometimes have basophils falling in here, you can have some other cells, and you can also have debris coming in there. So this is the region which is defined as the blast region. Now इसमें क्या दिख रहा है? These granulocytes and blasts are wide apart. टूट रहा है ना? But in this gradually going into your granulocyte gate. So can you tell me किस acute leukemia में ऐसा होता है कि blast से कुछ maturation हो रहा हो? It can be a case of AML with maturation. ये blast भी हो, mature भी हो रहे, more than 10% आ रहे हैं. So you type of getting there. The blasts are coming in the dim region, but they are gradually going into the negative area. So you have to. Now this is another plot. What I told you is blast will come in a region which is dim 45 and relatively lower side scatter. But what here it is happening is the blast here are going up and they are having the higher side scatter. So this is a plot of a case of APML. APML में क्या होता है? Promyelocytes होते हैं. So they have a higher side scatter. So this is a case from a APML. Then you see here. Normally, we are expecting blast to be here, but now you can see the blast are sitting. Normally, monocytes are there. So this is from a case of acute monocytic leukemia. So see, you cannot see the blast with your eyes on the flow plot, but if you know these patterns, you can probably link it to the morphology. And obviously, it's best to see the morphology before to the plots. So this is again, this is the case when I told you that sometimes the blast can be negative. Now you have processed the sample, you see lymphocyte है, granules होगा, शायद RBCs होंगे वो. These cells, you might miss getting these cells, but if you get these cells and see what they're expressing the immaturity marker 34. So you can say that yes, blast here in this case are negative for 45, but they are blast. So the conclusion. Blasted in 45 and low side scatter, but depending upon the it will be negative for 45. So we have seen to identify the blast, but does flow is the method of choice to enumerate blast? No. Still morphology is the superior one. Flow has not been given the role of assessment of the number of blast cells. Then the next is you have identified yes, ये मेरा blast region है, इसमें कुछ blast जैसे cells लग रहे हैं. So now next step is you have to confirm that yes, these are blasts. So how you confirm? You will see whether they are expressing markers of maturity. Euroflow ने तो 34 को ले लिया है ना, but in addition you have 34. 30 is कि कोई lineage का marker आ रहा है कि नहीं. So you have CD19 here coming here. So these are blasts are positive for 30. हो गया ना डिसाइड कि बी सेल प्रिकर्सर एल की तरफ जा रहा है. Then in addition, what you can see is these cells are not expressing 20, they're expressing 34, not expressing 117, और इनमें 10 भी नहीं आ रहा है. ठीक है. So ये नॉर्मल, ये यहाँ साइड में क्या दिख रहा है? These are the maturation pattern of normal in C which mature from stage 1, 2, 3 and finally go to the mature B cells. So ऐसा पैटर्न तो हमें दिख नहीं रहा. हमें क्या दिख रहा है? सिर्फ एक बटन दिख रहा है. अगर इसमें नॉर्मल सेल्स होते तो इस तरह से हमें कुछ सेल्स इस जगह पर मिलते, कुछ सेल्स यहाँ मिलते, कुछ सेल्स यहाँ मिलते 10 और 20 के प्लॉट में. यहाँ पे तो हमें we can't find it. So this is a then what you will see is you will apply additional treatment. सिर्फ एक, जैसे अगर हमें लग रहा है स्लाइड से देख के भी हमें ALL ही लग रहा था ना, क्या हम ऐसा कर सकते हैं? सिर्फ B के मार्कर लगा के कर लें. You have to put in all the markers because you have to provide a complete immunophenotype. तो इस केस में we can see there is some expression of ये green. The green ones were normal granulocytes. तो हमें देखना है 1333 हमारे ब्लास्ट में आ रहा है कि नहीं आ रहा है. तो हम पहले किससे देखेंगे? हमारे ग्रैनुलोसाइट्स में आया है कि नहीं आया. दिस विल हेल्प यू चेक कि उससे हम इंटेंसिटी भी कंपेयर कर लेंगे. अच्छा हाँ मतलब अच्छा So here you can see that ये चल ही रहा है, ये नहीं चल रहा, ये दिखा नहीं रहा है मेरे उस पे लाल हरा इस पे वो मेरा लेजर पॉइंटर भी हैंग हो गया, वो भी हैंग हो गया। हाँ ये नहीं वही है, मैं वही चिपका ही हुआ है, उसको शो ही नहीं कर रहा है, नहीं उस पर शो कर रहा है पर वही चिपक गया है, उस पर नहीं। राइट क्लिक करके आप उसको पेन पे कर दो, पेन पे सीधा स्लाइड आगे बढ़ेगी तब पॉइंटर भी ना फ्लैट हो गया। अब इसको स्लाइड शो को एंड करके फिर से चार से अब वाली ग्रैनुलोसाइट्स है ना, हरे वाली जो है, तो इसमें 13 आ रहा है ना, और 33 भी आ रहा है, और हमारे ब्लास्टर कमिंग टू दी सेम पोजीशन जहां उसमें 33 आ रहा है ना, इसमें 33 यू विल सी लाइक दिस, ये 33 आ रहा है ना ग्रैनुलोसाइट्स में, आपके ये ब्लास्ट जो रेड पॉपुलेशन में थे इनमें भी उतना आ रहा है, 13 के हो गया 33, ठीक है। So then further you can categorize on flow cytometry कि BLL है तो वो प्रो भी है, कॉमन भी है, प्री भी है या मैच ये वाला है, ठीक है, डिपेंडिंग अपॉन द एक्सप्रेशन ऑफ 1910 दी साइटोप्लाज्मिक। देन व्हाट आई टोल्ड यू कि एक था कि ब्लास्ट हमें आइडेंटिफाई करने थे, कंफर्म करने थे, लीनिएज असाइन करने थे, तो नेक्स्ट वाज कि हमें कुछ साइटोजेनेटिक ग्रुप्स या कुछ यूपी में एसोसिएटेड इम्यूनोफिनोटाइप बताने थे। So for the Euroflow panel, it includes these markers 22, 24, 66C. This is a list identifying the leukemia. Then the flow also helps you to tell the immunophenotype and genotype correlation for BLL. So supposedly you have this translocation 922 wala, so it will be expressing your 25 along with aberrant myeloid markers like 1333. Then if you have this, so what you will have, you can have that will be generally negative for 10 and it will be expressing 15. So this is just a table. You can remember, you just remember कि flow से we can also tell the correlation with the cytogenetic abnormality which will be there in the patients. Now this is again granulocytes and lymphocyte and monocyte, and these are your blasts, which are 45 dim and they have a low side scatter. Now this blue population is your normal lymphocyte. So first we'll see what is coming in your normal lymphocytes. Whenever you start interpreting the flow plot, First see whether all your markers are granulocyte कितने B कितने. Then go to the blast population. So that will help you analyze it better, मतलब easily. So इसमें हमारे 7 और 2 आ रहा है, फिर इसमें surface CD3 भी आ रहा है और 7 भी आ रहा है, और इसमें 5 भी आ रहा है, और ये थोड़ी थोड़ी population 4 है, कुछ population 4 और 8. Now we'll come. इसमें B और माइलॉइड के नहीं थे, तो हमने सिर्फ T के लिए दिखाए आपको. तो इसमें now this 7 is coming to the similar intensity as it is coming to your normal lymphocyte, है ना, वो भी यहीं आ रहे हैं, ये भी यहीं आ रहे हैं, पर ये नीचे गिरे हुए हैं. क्यों? Because they are not expressing CD2. They are only expressing marker का loss हो गया blast. ही आया. Then the next thing is you can see here these are your cells which were bright for 3, and again your blasts are not showing what? They are not showing surface CD3 expression. ये जो cells है इनमें 7 आ रहा है, पर surface जिसमें आपको 5 ना मिले तो आप सोचिए कि हाँ ETPLL के लिए. So you'll have your markers, and in addition, so just for this case you can see again ये 33 in blast में आ गया similar intensity में, जिसमें granulocytes आ रहा है. Case of ETPLL positive पर जिसमें 180 नहीं आया, and there was 34 heterogeneous और बाकी कोई markers नहीं आए. ठीक है. TELL, you can classify it as pro-T, pre-T, cortical T, mature T, but then you have to have the whole marker, whole list of the markers. For what we see here is for ETPLL it is 7 positive, 1 A negative, and 5 is usually negative, 34 positive, and you can have these myeloid markers. Okay, so बाकी आप categorize कर लेते तो बहुत अच्छा है, but what flow needs is that you must tell the ETPLL where, है ना. Coming to the क्या है, TELL में अगर इमेच्योरिटी मार्कर आ रहा है कोई भी आपका, तो दैट इज द ल्यूकेमिया एसोसिएटेड इम्यूनोफिनोटाइप. कि नॉर्मल ब्लास्ट तो होंगे ही ना. टी के ब्लास्ट कहाँ होते हैं नॉर्मली? टी के ब्लास्ट टी सेल्स कहाँ मैच्योर होते हैं? थाइमस में होते हैं ना. तो बोन मैरो में अगर आपको टी के ब्लास्ट मिल रहे हैं, इसका मतलब है कि दे आर एबनॉर्मल ओनली. So now this is another case. आप इसके आप देखिए, अच्छा इसमें मैंने वो प्लॉट नहीं लगाया, सॉरी, मतलब आई जस्ट वांट मिस दैट प्लॉट कि जब हम फॉरवर्ड स्कैटर साइड स्कैटर लगाते हैं तो ब्लास्ट में आते हैं. दैट दिस इज योर नॉर्मल लिम्फोसाइट, और ग्रैनुलोसाइट गेट में बहुत कम सेल्स हैं, और ये जो सेल्स हैं नॉर्मली हम यहाँ क्या लगाते थे, ये जो एम लिखा है यहाँ हम लगाते थे मोनोसाइट का गेट लगाते थे, और यहाँ पर कोई और सेल पॉपुलेशन आ गई जिसकी वजह से हमें मोनोसाइट को साइड कर दिया है, और इसी को हमने क्या बना लिया, दिस गेट वी आर मेड ऑफ. See this M is sitting ideal कि अरे मेरी जगह पे कोई और आ गया. ये कौन से है? मोनोसिटिक ब्लास्ट है. फिर जस्ट सी कि कैसे है. So for other leukemias what we say 34 TD 34 usually negative for 34 monocytic leukemias. Then 19 नहीं आ रहा, पर हमारे नॉर्थ में 19 आ रहा है. तो sometimes maybe that नॉर्मल सेल्स में आ रहा है कहीं, लिम्फोसाइट में 19 आना चाहिए, तभी आप कहेंगे कि ब्लास्ट में नहीं आ रहा है ना. देन फिर ये 10 और 20 भी नहीं आ रहा है, ये हमारे नॉर्मल में आ रहा है 20. अच्छा 10 के लिए क्या यूज करते हैं नॉर्मल में, इसमें दिखाया नहीं है मैंने, ग्रैनुलोसाइट्स यूज करते हैं, इसमें नॉर्मली 10 आता है. व्हाट दे आर एक्सप्रेसिंग इज दे आर एक्सप्रेसिंग ब्राइट 33 एंड हेटेरोजेनस 30. 33 ब्राइट क्यों, एक ही जाके सारे, और 13 में ऐसे स्प्रेड are acquiring 14 and then negative for 117. So what we classified as acute monocytic leukemia. So this is my last slide. This is just to show you again a case of APML. We were suspicious for APML, and then if you don't have the facility for urgent reporting for fish of transcription 1517, you can run a flow, and that will be almost equivalent only. So here you can see these plots are not from my lab. These have been taken from a paper. You can see that this is a population which is showing high summer lymphocytes. Bright 33, then they are showing 117, and they are negative for 34 in HLA-DR. So for APML, what we say is you will have a region, you will have a population which will have a higher size content in 45, and that will be negative for 34 in HLA-DR. 17, and obviously they'll be bright positive for MPO. So again, the flow here is helping you find out an association with the cytogenetic or molecular abnormality in the patient. And in the last, I'd like to thank my teacher, late Professor Ashutosh sir. So are we having questions here or? Okay. Thank you. Yeah.
डिवाइस का firewall
ब्राउजिंग
ब्राउजिंग ब्राउजिंग
वाईफाई इंटरनेट का करना था कर दिया ना
और विंडोज का भी firewall
इसके अंदर का
विंडोज में firewall होता है
हाँ वो तो होता ही है
उसमें firewall
अच्छा इन्होंने टीवी कल ऊपर लगाया तो पूरा सिस्टम ऊपर कर लिया ना लग रहा कपूर साहब ने नया खरीदा है
और बैक बैक करके सारे
पब्लिक आ रहा है प्रेजेंटेशन अभी हटा दी देखो ना कैमरा देखो
चेक कर लो
आवाज आ रही है आपको
Presentation.
presentation मैम बोल दो presentation करा मैं आपका क्या presentation conference
इसमें से ये सेकंड वाला
दोबारा एक्सेप्ट कर दीजिए
एक्सेप्ट है
Okay, so I will tell you in brief how to approach the chronic lymphoproliferative disorder, the B cell type. So as we all know that B cell NHLs are mature B cell neoplasm. Okay, you have B lymphoblastic leukemia that is immature, and then you have B lymphoproliferative disorder, and it's a general concept that tumor cells tend to recapitulate the stages of maturation. As you know that of maturation, antigens they express at the various stages of maturations will be to a certain extent maintained in the B cell mature neoplasm. At times they will be lost or they will be altered, but to a larger extent it will be maintained. So how do you proceed? You get a patient with increased count. So you take the history initially, you do the clinical examination, check for the is very important. You cannot escape that and you should not skip it. Take the detailed history, the mode of onset of the symptoms, then do the complete examination of the patient, check for any organomegaly, splenomegaly, presence of any lymphadenopathy, how many lymph nodes are there, and then we proceed ahead with blood smear. For the count, in the smear we evaluate the type of cells present. Then we do bone marrow aspiration and preprime biopsy. Bone marrow aspirate will give you a clue to the larger extent the kind of hemopoiesis or the infiltration in the marrow that has taken place, and further which is required to know trabecular, intratrabecular, all those things you'll get to know and refine. Then we proceed ahead with the immunophenotyping with the help of flow cytometry, and further we go ahead with the cytogenetic and molecular. So this is the B cell, normal B cell maturation that I was talking about, pan B cell marker, and it gets expressed throughout all duration stages. Then you have a pan B cell marker, but look at CD20. CD20 is almost absent in the very immature stages, starts getting expressed in the pre B cell, and then it gets expressed in the memory B cells. Okay, and then we have other B cell markers also, like you have a BCL6, CD38 in the plasma cells, and many other markers. But CD19 is a pan B cell marker, and so we'll use CD19 disorders. This is a newer classification, and this article is available on the internet. You can access it. This article has shown the changes which the newer classification has undergone. The new entities that are included and the older entities that has been removed. Just put it in has been removed. In situ follicular neoplasia is called as in situ follicular B cell neoplasm now, and there are many changes. I am not going into the detail of it. Okay. Now, what are the various samples that you can run in the flow cytometer? You can use the peripheral blood, you can use the bone marrow, you can use. So, if there is no marrow or peripheral blood involvement, you can, and if the lymph node is involved on FNAC, you can take that FNA material and run on the flow cytometer for further subtyping. You can use a body captive fluid if the patient is having ascites or you having a pleural effusion or if there is infiltration into the CSF, and sepsis by grating it through the special meshes which are available and suspending them into the liquid media, and then you can use them. So how do we approach? First of all, we should be sure whether it is a lymphoproliferative disorder or not. Then whether it is B cell, T cell or plasma cell disorder, that you will get to know with the help of those. Markers if they are available for the targeted therapy, and as well as to check for the presence of any prognostic markers. Now this is the basic panel, 8-color panel that we use in our lab. As you can make out, CD19 is being used as a gating marker. CD19 marker has to be put into all the tubes. All B markers, the next tube has clonality tube with kappa and lambda, and then we have a tube to determine for the presence of, to check the presence of hairy cells. And further, you can add a new panel. So lymphoid cells are graded on CD19 versus site scatter. Acute leukemia we used to use CD45 versus site scatter. In BCLPD we prefer to use 19 versus site scatter. Okay. And usually on CD45 plot, these B chronic lymphocytes. This is a B lineage. As you can make out, we are using CD19 versus site scatter as the gating plot. The 19 positive population and there is a 19 negative population. So your 19 positive cells are the B cells, and then you have the 19 negative cells which are the T cells. Using 19, that patient is not on certain therapies like CAR T cell therapy or anti-CD19 like blinatumomab, because at that time the cells will be having loss of CD19. So you have to take the history completely before putting a gating marker. CD20 is not used as a gating marker, but because you saw that it gets expressed only after initial certain stages of maturation. Second, in certain NHL patient is given anti-CD20 therapy like rituximab. So we don't prefer to use 20 as a gating marker. In certain B cell neoplasms like CLL or SLL. Now, how do you identify whether the B are normal or abnormal? So that can be done with the help of immunoglobulin light chain class restriction or the presence of usually not expressed. The antigens which are usually not expressed by that lineage, and if they get start expressing in a particular lineage, like CD5 is a T cell marker. It should not come in the B cells. But if your B cells start expressing CD5, it is abnormal. So this is being expressed. Suppose there is CD20 and there is loss of CD20 in certain cells, in B cells. Then you know that your population is. The next thing is immunoglobulin light chain. The B cells, some of them are kappa positive and some are lambda positive. Okay, you can make out from this plot a kappa positive. This is called as a reactive population or polytypic. Some cells are kappa positive, some are lambda positive, with a ratio of 1 to 2. But if in certain case or in certain population you so that becomes monoclonal, or all cells starts expressing lambda, it becomes monoclonal. So this is how you check for the clonality. Complete loss of lambda expression and the whole population is positive for kappa. So you monoclonal population positive for to know that you are dealing with an abnormal population, and then you check for the other markers getting expressed. Some B cells can be light chain dim or negative, like CLL. CLL will not have a very bright expression of kappa or lambda. It will be either negative or about that. And in certain body fluids, if you are processing, the B cells might have dimmer expression of clonality markers. The germinal center B cell usually have a dimmer expression of these markers, and the blast and hematogens also have a Last set expression of these. Okay. And not every monotypic population is lymphoma. Every time you see a monoclonal population, don't jump to a conclusion that okay, it is lymphoma. It's not always true. Especially in cases of monoclonal B-cell lymphocytosis or in situ follicular neoplasia, especially in this monoclonal B-cell lymphocytosis, although the population will be monoclonal, but this you have to be sure. Like in HIV patients, in which you get reactive lymphoid hyperplasia, at times you can get a clonal expression of either kappa and lambda in those cases. The expression is not enough to call other markers as well. This is about the aberrant antigen expression, which I have already explained. Expression of those antigens that are normally not expressed, like expression of 13 or 33 by B lymphoid. You know 13 and 33 are marker of the myeloid lineage. If B cells start expressing them, it's abnormal. Similarly, expression of 5 on B cell. 5 is a T cell. Similarly, expression of BCL2 on 10 positive cells. CD10 is a marker of germinal center. Okay? So CD10 is a center and BCL6 center. Whereas BCL2 is expressed by the cells in the periphery, in the mantle zone cells. Okay. So if your 10 positive cells start, okay. What are the normal pattern of expression of markers? Like your germinal center is positive for CD3 usually comes in the interfollicular area, but in the germinal center, some cells will be CD3 positive, which are T follicular helper cell. So germinal center will have some ammonia expression. Okay. Now, how do you assess the size of the cell? As I told you in the first lecture, give peripheral smear keratosis and the CLL, the most likely. Okay, and this is the plot. As you can make out, the cell size is very small. So these cells will come somewhere here in the forward scatter as well as very low on the side scatter. So this will give you a clue that you are dealing with a population which is very small in size, very compact nucleus, no cytoplasmic granularity because very low on side scatter. Okay. You are seeing slightly bigger cells. There are some cytoplasmic irregularities, some projections. So let's look at this plot. This population is further ahead, slightly larger on the side scatter as well. So your forward scatter versus side scatter, to a certain extent, it will give you a clue about the kind of cell you are dealing with. So this is a case of hairy cell leukemia. Even in Burkitt's, the cells are large, so population will be slightly forward and higher on the side scatter. Now look at this population, slightly larger, even larger lymphoid cells you are seeing here. So what on this side scatter and on 45 versus side scatter, this population is coming and lying above the lymphocytic region. So this is B-cell lymphomas. Exactly in the region of the mature lymphocytes and slightly above them in the monocytic area. The abnormal population lying in the lymphoid cell. Think about all the lymphomas which are having presence of large lymphoid cells like diffuse large B-cell lymphomas. Okay, or okay, even at times T histiocyte rich B-cell lymphomas. Okay, so this is an article. It is an old article, but still very helpful for and gives a basic approach by Craig and Kuhn on the hematology. I'll be talking based on this article. So we classify mature B-cell neoplasm into these 4 categories based on the expression of CD5 and CD10. 5 most common CLL bar SLL, mental cell, PLL, marginal zone, at times DLBL. DLBL can be 10 positive or 10 negative. Then you have 5 negative, 10 positive. So 10 positive means all the lymphomas arising from the germinal center. Here you have follicular lymphoma, diffuse large B-cell lymphoma, Burkitt's lymphoma, and 5 and 10. Here you have hairy cell, most common, then marginal, 10 negative diffuse large B-cell lymphoma, and very rare cases of follicular. Follicular is usually 10 positive, but very rarely can be 10 negative. And then you have 5 positive, 10 positive. Both positive lymphomas are follicular, DLBL, mental cell. Can use, or you may use this approach also. both the approaches are very beneficial. Like check for the presence of CD5. Is CD5 is positive? Check for the presence of 23 and FMC7. Now is negative, it is CLL. 23 positive, FMC7 positive, it is pro-lymphocytic leukemia. If 23 is negative, FMC7 positive, it is mantle cell. Now, if 5 is negative, check for the presence of CD10. If CD10 is positive, you are dealing with the all the molecular like B-acute lymphoblastic leukemia. If 10 is negative, now check for the presence of all the markers for the hairy cell. If you are seeing the cells with hairy projections or irregular cytoplasm, here you have 103, 125, and 1 is hairy cell leukemia. If negative, it could be either of these. So you have to check for the presence of the other marker. So you can use either of the two approaches. Now this is case 1. Just to show you a few cases how the approach is done. 60-year male with leukocytosis and lymphadenopathy. ठीक है, ये peripheral smear very classical क्या है CLL. Singlet cell gating you will learn in the subsequent hands-on after the lunch, and you can this is forward versus side scatter. We have gated a 19 positive population. Okay, your population is 19 positive. Now check for the clonality. Some cells there is no lambda expression, so you're dealing with a clonal population. Okay, and the cell size is small. Check for the other markers. You can make out heterogeneous का मतलब क्या होता है? Some are dim positive, some are negative. This is all heterogeneous. Bright positive, if you get a, you call it as bright positive, you get a clear-cut button here, or negative if you get a clear-cut button here. Okay, one more thing I wanted to tell you. When always look at the normal internal control. ठीक है, तो यहाँ पे आपने एक गेट यहाँ पे भी लगाया ये P2. P2 is what? 90 negative cells are what? T cells, T lymphocytes. ठीक है, तो 90 negative region में जो lymphocytes आए आपके, those are the T lymphocytes. So look at the subsequent plots. On this 23 abnormal population is coming here. So 23 is heterogeneous, 38 is negative. Look at 5 and 45. Both are bright positive. Okay. These are your T lymphocytes which are 5 positive. Okay. Now look at CD20. CD20 is again heterogeneous. Right? I hope you CD200 is what? What kind of expression it is? Right, है ना? ठीक है, क्योंकि देखो ये आपके T lymphocytes हैं. If you compare with this, you are getting a button here. So 200 is bright positive. But look at 20. 20 is heterogeneous. Okay. What about 4 and 8? Both negative. What about CD7? CD7 and 3 both are again negative on this population of interest which you have gated, and these are the normal T lymphocytes. So you have a here of small size cells and with a bright expression of 200, heterogeneous expression of 20, dim kappa, FMC7 negative. तो क्या है ये? CLL will have a dimmer 20 and 200 is very brightly expressed in CLL and in hairy cell leukemia. In certain other lymphomas, it can be expressed, but to a very lesser extent. So CLL is very bright 200. PLL, BPLL will be 200 negative and 20 bright. And another small cell lymphoma is mantle cell lymphoma. So mantle cell lymphoma is 5 positive, 20 positive, and will have expression of cyclin D1. CD200 and 23 will be negative in mantle cell lymphoma. So these three are small. Each one of them. This is case 2, 53-year male with weakness and splenomegaly under evaluation. You can make out the bone marrow. It's infiltrated by all abnormal lymphoid cells. You are not seeing the normal precursors. So again, kappa positive. So you have a monoclonal population being gated on CD19, and look at the other markers. What about 5? 5 क्या है? Heterogeneous, ठीक है. 23 negative. bright, 20 bright, 38 नहीं नेगेटिव नहीं है, 38 is on this axis, so this is positive. ठीक है. This is case 3, 52-year female with dry cough and chest pain. Follow through case of pancreatitis with 8 centimeter palpable spleen. With this patient is a TLC, and in fact there is pancytopenia, and most of the cells are lymphoid cells here. Certain lymphomas do present with pancytopenia, and you must know about them. ठीक है. Okay, so this is a plot. You have been positive for. So 23 is positive. What about 200? 220 bright. And because of the parafibrosis, these patients can have diluted bone marrow aspirate. And these are the markers: CD25, CD103, CD123, markers for the hairy cell leukemia. Okay, these are the. So this was 5 negative, 10 negative, probably hairy cell leukemia. Now, just I have put these two pictures of splenic marginal zone lymphoma, or probably this kind of bipolar sections are seen in this. And there are certain cells, if you see projections with prominent nuclei, those are seen in the hairy cell leukemia variant morphologically. And how to differentiate between them? Hairy cell leukemia variant is 25 negative. Okay, it will be 103 positive, 200 negative, 25 negative. Whereas SMZL will have 305. So this table gives a gist about how to differentiate between various lymphomas using the hairy projections. Okay. This is another case, 33-year male with recurrent vomiting and weight loss and pain abdomen. You are seeing these kind of cells here. CT abdomen showed heterogeneous lesion in the third part of the duodenum. Okay. These are various cells. What can you tell about the size? They are large cells because they are 2 to 3 times larger than the RBC. So you know you are dealing with the large. Okay. Look at this plot, 45 versus side scatter. Your abnormal population is coming on top of the mature lymphocytes. Much higher on the forward scatter and slightly higher on the side scatter. These cells are bright 19, gated on 19. There is expression of CD10. So large cells. Germinal central lymphoma. Okay. 38 is positive, 20 is bright positive, 5 and 2 negative, 200 is negative, and lambda clonality. We have a 5 negative, 10 positive lymphoma. So if you talk about large lymphomas with 5 negative, 10 positive, you have diffuse large B-cell lymphoma. But I have kept other two differentials, follicular and Burkitt's, just keeping in view the immunophenotypic pattern. This Burkitt's lymphoma and follicular lymphoma have a very characteristic morphology. Like follicular lymphoma, they have small cells with nucleus. These were the with history of weakness, joint pain, abdominal discomfort, palpable spleen and liver, and retinopathy. Almost 1 to 1.5 times. There is a nice nuclear cleaving. Can you make out here? Okay. Can you make out this kind of nuclear cleaving? Right? In almost all the cells. Okay. Now, this case we gated on 40 side scatter. Right, another one slightly dimmer. So you are like, whether it is a plastic lymphoma, we are dealing with acute leukemia or mature. So we use 45 as a gating marker in this, and further saw that these are expressing 10 and 19, 20 is bright, 34 is absent in this case. We had done a myeloid tube also. All the myeloid markers are negative. HLA-DR is getting expressed. HLA-DR is also a marker of activation. Okay, and another markers like cytoplasmic MPO is negative, 22 is positive. Kappa lambda. In the case of follicular lymphoma, 34 is negative. B cell markers are positive. Morphology is characteristic. Small cells with nuclear cleaving, and kappa and lambda is negative. Why? Because of the procedural errors. when you are processing the combined lambda tube, you have to be very careful that you have to check initially. Non-specific binding of the immunoglobulins. So, ideally, when you are proceeding with the combined lambda processing, take the sample in the tube, wash it 3 or 4 times, and then proceed ahead with the staining. If you don't wash it properly, you will not get a proper chronality assessment. And markers by various neoplasms, B cell neoplasms. So take home message is diagnosis of BNHL is combination of history, morphology, immunophenotype, and some cases requiring the cytogenetic and molecular assessment. You can get a clue about the size of the cell based on your reliable gating marker is CD19. Then check for the clonality markers like kappa or lambda, which will help you in telling whether you are dealing with a monoclonal or polyclonal population. And just remember that the tumor cell always recapitulate the various stages of maturation of the normal B cell. So remember the normal B cell maturation and the markers. Please check for the presence of aberrant markers or altered intensity of markers. Thank you. Now I would like to invite Dr. Jasmita for T chronic lymphoproliferative disorder. Yeah.
So, how many of you are awake? Nobody. How many of you are awake? I'll ask the question again because no hand went up. All. Okay. So great. I hope you enjoyed the period. So we'll talk about TCLPDs today. So for BCLPDs, you have a nice gating marker in the terms of CD19. For T-cell lymphomas, if you actually remember your T-cell lymphomas or acute leukemia, you had cytoplasmic mandate present in all the cells. However, can frequently you can have T-cell lymphomas that are surface C negative. You can have T-cell lymphomas that are CD7 negative. So we can't start with one gating marker alone. We have to look at the T-cell markers in total. So if you remember, Millie would have shown you the approach, the Euroflow approach, in which we orientation tube and then the lymphoid leukemias also for mature lymphoid ones, as in the lymphoma phenotyping. You again have lymphoid screening tube followed by B and T further tubes. So lymphoid screening tube has a combination of different markers. So it has CD4, CD8, CD3, CD56. Lets you identify the NK cells. Along with that, it has CD5. So there is a population that is CD5 positive but surface CD3 negative, not expressing B markers. That also becomes a suspect abnormal T cell population. Similarly, when you actually look at the rest of the markers, it will have 19, 20, kappa, 8. Okay, so you know, told you already. So we started with 3, 5, 4, 8, 19, 20, kappa, lambda, 45. 38, and what else? PCR gamma delta. So if you count these markers, there are 12 markers. So how do you put them on 8 colors? Question, right? On your equipment limits the number of markers you can use. But remember, you have two distinct populations, that is the B cells and the T cells. So T cells will not express kappa, lambda, right? They will express 4 and 8. So there is an option of multiplexing. So in the same fluorochrome, you have kappa, say when to analyze them on different cell populations. So that is an example of multiplexing, which you will see in my presentation, because we use the approach where we have a lymphoid screening tube, followed by that we put a B or T, dependent on what we get in the lymphoid screening tube. So for TCLPDs, and I will not talk about NKCL today, what we do is that we take
CD3 positive, surface 3 negative, exclude NK with the caveat that lymphomas can be CD3 negative. So you may have to start with CD7 or CD5 as the alternate markers. So in the category of dual positives, that is CD4 positive, 8 positive, you have one in practically none. This entity is also common in cortical TLLs, right? The ones which have CD1A expression. So T lymphoblastic lymphomas and leukemias you have to exclude when you're talking about a double positive population.
Then you have the CD4 positive T cells. We encounter our TPLL, mycosis fungoides and Sézary. So mycosis fungoides, Sézary is which kind of a T cell lymphoma? Nodal or cutaneous? So primarily a cutaneous involvement, right? Then the other entities are acute adult T cell lymphoma leukemia in our country. See this is not at all. Then you have the angioimmunoblastic T cell lymphoma. This is one lymphoma, the recognition of which is actually increasing because of the increasing number of markers being applied and increasing recognition that this entity exists. Then rare cases of TLGL, ALCL, cases of PTCL can also be there. CD8 positive lymphomas which are 4 negative. So 8 positive lymphomas are more of the cytotoxic phenotype. So you have the large runner lymphocytic leukemia. Some cases of TPLL, the hepatosplenic T cell lymphomas can express CD8. Similarly, the enteropathy associated lymphoma and subcutaneous paniculitis. Now, how frequently when you get SPTCL to flow? You will not get it if you are doing only peripheral blood and mass. You will only get it if you are analyzing the mass that they produce. So only the cutaneous lesion. Otherwise, no. Then in the double negative group, you have hepatosplenic T cell lymphoma, anaplastic large cell lymphoma. Now, anaplastic large cell lymphoma is one lymphoma where all the T cell markers can be negative and yet it is a T cell lymphoma. So that is again one thing which we need to remember. So how do we assess clonality? In T cells very clearly by kappa and lambda, and you should of kappa and lambda. Analysis of clonality was very difficult using flow cytometry. So you have expression of TCR alpha beta or gamma delta receptors on the T cells. However, normally also you have alpha beta positive T cells and gamma delta positive T cells. So if your gamma delta T cells become 80% of your T cells, very like a malignant population, but with the alpha beta positive population. So there was B beta analysis, which was a huge component of 8 tubes that have to be put up, very expensive. Only an occasional lab in our country was doing this assay. Otherwise, clonality traditionally is done by assessing the TCR gene rearrangements. Now there is a TRBC1 antibody. You have surface 3 expression along with the TCR alpha beta or lack of gamma delta. I'll show you examples of that as well. So with that, we'll quickly move to cases. So this is a 52-year-old male with fever, easy fatigability. The patient has splenomegaly. If you look at the counts, there is a expansion. So this is the morphology, classical morphology of large granular lymphocytes. So these are cells which have granules in them and they are lymphocytes. And if they are expanded, the entity you worry about is the LGL. However, remember that many patients can have LGL expansion. So if you look at your peripheral smears and try to identify them not just in LGL leukemias but in a host of other conditions as well. So I'm showing you these plots only once. These are my housekeeping plots. So we do forward scatter area versus time to take care of fluidics. If they're rapid fluid, like you have cells which are stable here, if they fall off, then you know that fluidics was a problem and you can only get the continuous expansion here because everything is okay, we'll get it. Area versus width has been used here. Earlier expansion. So remember the pulse height, you had doublet passing through, you had the first pulse going through interrupted by the second pulse. So the height remained constant, but the area and the width increased. So that is what we are utilizing. But there has to be a linear between area and height. Why? What did he do? So he changed my pointer options and left. 45 versus site starter plot. You can identify a population of lymphocytes. These are the maturing myeloid cells, and you see something else. So the minute we get the NDC, you realize that all this junk and clutter is gone. So this clutter and debris will complicate my further analysis. I can get rid of it upfront. So we get on the lymphocytes. Okay. Starting gate for T cell lymphomas that I prefer using is a lymphocyte is encompassing the puno region as well. Right. So we are trying to get any have a moderate site scatter or can have a. So that is where we start from. So then the next tube is 3 versus 19. So what do you see here? CD3 is much more than CD19. So we get CD3 positive cells as T cells. We get 19 positive cells as B cells. What else will be left here? NK cells, and because we get it monocytes, very likely monocytes also. Okay, so we have, if you see here, lambda is multiplexed with 56, right? Why? Because 56 positive cells don't express lambda. So I can multiplex these two markers on the same fluorochrome. So if you have a actually your NK population. So you've identified all the cells correctly. Simultaneous cells, the ones that express is not expressing CD56 to a large degree. So your case is 56 negative. What about B cells? When you look at kappa, what do you see? It's a nice split. There appears to be slight lambda expansion, but not enough to call it a malignancy till now. For 20, there is a bright expression. And then you go to the T cells. Can you identify some abnormalities? Starting from the first plot, 5 and 3. So what about positive, which is what you expect? But you have a population here that is completely negative for CD5. So loss of CD5 expression, and this is a huge chunk of the population. The same way you can see that you have the 5 negative population is expressing sorry, not kappa and lambda, but 4 and 8 because you are assessing T cells. So 4 and 8, if you see, what seems to be more, 8 positive or 4 positive? Cytotoxic or helper, which is dominant? Let's see. So we have two populations. We get them separately, the 5 positive cells and the 5 negative T cells. So in the next plot, you can clearly see that the 5 negative cells are expressing 38, brighter intensity. And what about 4 and 8 expression on this blue population? Clearly CD8 positive, right? It is expressed over a continuum, but these cells don't express CD4. They are all CD8 positive. So cytotoxic T cell expansion, CD5 negative with a large granular phenotype. And if I split these two to you nicely, you realize that around 98% of these are CD8, right? There is a massive. What about the other markers? So 56, you remember from the plot of NK T cells, initially only I told you 56 was negative. So here 16 and 56 are combined on the same fluorochrome. So you know that 56 was negative in the first tube. So what you are looking at is 16. What about 2? Would you call this 2 positive? Let us compare the intensity of CD2 between the pink and the blue population. 2 appears to be slightly, right? Intensity here, these cells, the pink cells, they are hitting 10 to the power 4. These cells, they are definitely lower than that. So 2 intensity is down-regulated. What about CD7? They're nicely expressing 7. And then 16 was done separately as well. So you know that 16 is positive. And you know that 57 is positive in the blue population, right? So this is the summary of the phenotype. So we have an expanded cytotoxic T cell population which expresses CD16, expresses CD57 in a subpopulation, shows a dim to absent 5 and slightly dimmer CD7. So with this, the bone marrow also came. So we assayed the bone marrow. Nearly same phenotype was available in the bone marrow as well. So this patient was suspected as a case of PLGL leukemia. Now, very frequently, you will have marrows with PRCA. This is one PRCA that we got. So, PRCA marrows, we usually do the T tubes because we don't want to miss a TLGL. So, in this patient, grossly, when you see this, you have the same B cell, T cell, NK, NKT, no expression of 56 in this. Again, 4 and 8, you look at this. There is a nice CD8 positive expansion. So, definitely, 8 positive cells are more than the 4 positive cells. So we move to 5 and 7 and suddenly we realize the problem that there are cells which are 5 dim, 7 negative. So T cells they are showing a loss of 7. So let us get these cells separately and we start seeing it. So CD2 is nicely positive, 5 is dim, no expression of 25 and these cells are again CD8 positive. So the 8 positive cells that were expanded are showing dimmer 5 and they are showing a loss of CD7. So these cells, you clearly have it here. And these cells, they express 57 in a majority. They don't express 16. And the NK was already negative. So with this, we thought that we have a TLGL leukemia again, which is the cause of PRCA. So therefore, identifying these small clones becomes very important. Confirmation is required for clonality. Now, this was a surprising case where we had a suspicion of a MDS-MPN. The patient had anemia and